So in this video, we're going to look at DNA replication in some amount of detail. I have a couple external videos that will give you a little bit different perspective on this, but I'm going to give you a slightly different perspective on this, highlighting some things which maybe don't get emphasized as well as I would like in other treatments. So let's get started. So here's going to be our workspace. Now, we know that DNA is double-stranded, and normally it is paired up so that we have AT base pairs and CG base pairs throughout there. And again, that's very important that the DNA stay in that state for as much as possible because it's a protective measure for the DNA, helps reduce chemical degradation and all that. However, you can't work with DNA when it's in this stable state here. So anytime transcription is happening, or in this case, DNA replication, we have to unzip the molecule here. So we're going to look at this from the perspective of a linear chromosome, although in prokaryotes have circular chromosomes. So their DNA would look like a circular piece, double-stranded circular piece. So that would be more like a typical prokaryotic genome, and it would also be much smaller. Prokaryotic genomes are very small compared to, say, eukaryotic genomes. So prokaryotes have small circular genomes. Eukaryotes typically have large linear genomes. But I'm going to look at this just from the perspective of a eukaryotic genome. Now, this genome is actually quite large, and if you tried to tackle it at one spot, it would take a very long time. So we're actually going to tackle it from multiple spots, but I'll emphasize that more later on, how exactly we do this from multiple spots. So first of all, one first thing that has to happen is the DNA has to unzip. And I'm going to represent that with a replication bubble. So this bubble that you're seeing here, this is called a replication bubble. And remember, we had something similar with transcription. Transcription also has to unzip the DNA, but there is an important difference we'll see here in a moment. But we had what was called the transcription bubble. Well, this is replication, so we'll call it a replication bubble. Now, this replication bubble starts out small, like the transcription bubble does, but then it's going to grow, and it's going to get much bigger, whereas the transcription bubble actually doesn't get very big at all. So, in fact, let me just show this as a series of images here. And then we'll go one more step here. So here's our replication bubble getting larger and larger, and eventually the bubble will, well, more details on this in a moment. It won't exactly be a bubble, but nevertheless, the entire original molecule that we start out with is going to be completely separated. Okay, that's actually where that's going to be the final step here, is that the entire molecule will be completely separated. However, during this whole process, we have replication happening, so we'll fill in those details here in just a moment. Now, the video that I have posted, the external video, only looks at half of the bubble. So let me clarify what I mean by this. So if you take a look at this replication bubble, you'll notice that there is this region over here, which we would call a replication fork. And this is essentially what's being shown in that external video that I have posted. However, it's important to note that there is another replication fork on the other side of the bubble. Okay, so in reality, there are two replication forks in the bubble. However, for simplicity or whatever reason, some people just show you one of the replication forks, because in fact everything that's happening at one replication fork is also happening at the other replication fork, just they're moving in opposite directions. So It is useful to just focus on one fork, but my personal preference is just to consider both of them simultaneously. So that's the process we'll go through. So I'm going to just go get rid of those boxes there. And then notice that the replication fork actually changes. It actually moves, right? So now our replication fork is here, and then later on the replication fork is here and here. So notice, in fact, I'm going to go ahead and put this back in here for just a second. Notice what's happening to the replication fork as we move. The replication fork itself, that one is moving off in that direction. This one is moving off in that direction. See that? This is very important, and you'll see why in just a little bit. But those replication forks are moving in opposite direction. You can think of this if you had like a piece of string and you made it and you doubled it over and then twisted it into a helical structure, okay? And then, well, you could do this with a rope too, right? A rope is already sort of like this. If you try to separate the strands, creating something like a replication bubble, and you open up those strands, you would see the forks as they are moving in opposite directions. And the bigger that bubble gets, the farther those forks move away from the center of that bubble. So we can think of this as kind of where the bubble started forming. That's the center line there. That's just an imaginary line there, but it's useful to have that guidepost there, as you'll see in a moment. All right, so an important point here is the replication bubbles are moving opposite directions away from that center, that imaginary line running down the center. Okay, so I'm now going to get rid of all those marks there and begin with, go back to our blank slate. Okay, so now this part's a little bit hard to imagine. Typically in class, I would pull up like an electrical cord. I would double it over and then twist it and create a double helix, essentially. And what will happen, you can even do this experiment on your own, or you can probably even visualize. It's not that difficult. But if you were to have a double helix and then you started separating the two strands and you pull those strands apart, what will happen is you'll get a buildup of tension in the molecule. You'll get a buildup of tension because this part of the molecule here, because it is helical, it will build up tension there and there, just beyond where the replication fork is. Now, again, my image here is, my original image is not helical, so this would not be true if it were not helical, but in reality, DNA is helical. So because of that helical nature, once you start to separate those two strands, if you were to grab this strand and move it up and this strand and move it down, you would get a buildup of tension. You would get tension here and you would get tension here, okay? Which is where my arrows here are pointing, right? Tension. That tension, that T there, that's called torsional strain. Very important concept. Torsional strain. Now that tension builds up very quickly, so by the time you got down to, say, this second or third or fourth image, that torsional strain could actually prevent the bubble from getting any bigger, because it's a resisting force, and it could actually prevent the bubble from getting any bigger. And if there wasn't a mechanism to release that torsional strain, DNA replication would actually come to a stop. It wouldn't be able to happen. All right. So we're going to come back to that in a moment, and we're going to talk about how to resolve that. Now, before we get to that, though, let's introduce the concept of a helicase. So there would be a helicase. I'm going to represent it with this blue circle. I assume it's blue. So that's called a helicase enzyme, and this is mentioned in that other video. helicase or DNA helicase. This enzyme rotates at a very high rate, and basically what it's doing is it's separating the two strands. All right, it's unzipping the molecule, if you will. Or another way of describing that is it's turning double-stranded DNA into single-stranded DNA. Okay, so that's what we mean by unzipping, or you will also hear the term melting of DNA. Melting does not have the same meaning as what we typically think of it as a solid turning into a liquid. Rather, it's a double-stranded molecule separating into single-stranded molecules. Okay? So that is DNA helicase. That's going to be the first, essentially the first thing that happens is DNA helicase attaches, two of them actually, to create this bubble, and they start moving away from one another. And again, those are going to be moving in opposite directions, just like we talked about before. All right, and that's going to cause this buildup of torsional strain. It's going to be a result of DNA helicase is what's causing that torsional strain because of the melting of the DNA. Now, we have to release that torsional strain, otherwise, again, things will not continue. So I want to use a different color here to represent a different enzyme. And again, I'm just going to use circles here to represent this enzyme. Essentially, there's an enzyme called gyrase, DNA gyrase. That's a common name which is used for it, but I also want you to be aware of this term here, topoisomerase. DNA gyrase is a type of topoisomerase. And I'm going to come back to that term here in just a moment because it's an interesting word. But let me first of all explain what this enzyme does. So imagine again that we have this torsional strain built up here and here in this helical molecule. One way to release that strain would be to cut the DNA and then allow the helical, that torsional strain that's in that helix, to just unravel, release that strain just naturally. Now, you could, in theory, you could cut one of the two DNA strands, and that would do it, right? So imagine you know, if you had like a rope and you tried to separate the fibers, you would get this torsional strain. You could easily release that torsional strain just by cutting one of those strands. Of course, we don't want to leave the DNA cut like that. If you do cut it, then you want to repair that cut. So then you would have to connect those two pieces together, but after that torsional strain has been released. So that's essentially what DNA gyrase does. There's different forms of DNA gyrase. Some of them will only cut one of the two strands. Some of them will cut both strands. The more common one that's used in eukaryotes actually cuts both of the two DNA strands. But then the same enzyme will repair its own cut after that strain has been released. So the animation in the video, the external video that I have linked, it shows this better than I can on these still images. So that reference will work quite well. So just imagine the two DNA strands actually being cut. The DNA, the torsional strain is released, and then the cut is repaired. Now, notice as the bubble gets bigger, that strain is going to continue to build up. So as our helicases continue to move farther and farther apart, that torsional strain will come back, and when it does, DNA gyrase will have to do its thing once again. And sorry there, I don't know if I think I actually, did I change colors? I don't know, but didn't mean to change colors there. Anyways, the DNA gyrase will have to repeat this job multiple times, okay? All right, so that's basically kind of the beginning of the process. We have this melting of DNA by DNA helicase. That leads to this torsional strain, which then requires DNA gyrase to come in and attach and release that strain. So why is this called a topoisomerase? Well, first of all, you should know by now that an isomerase basically is an enzyme that takes one isomer form and creates a different isomer form. So in this case, what's happening is the enzyme is cutting the DNA and then it's repairing its own cut. So it probably makes sense that in the end, chemically, DNA is not being changed, right? If you cut the DNA at a particular spot and then you repair that cut, it's almost as if you didn't do anything at all, except, in fact, something did happen. The torsional strain was released, right? So that's why we call it an isomerase, because it does a chemical reaction, but then it undoes that chemical reaction. So by the time it's done, by the time the DNA gyrase is done, chemically the DNA has not been altered. However, what has been changed is that torsional strain has been released. In other words, the topological features of DNA have been changed. So instead of having, let's see if I can draw this here. So here I'm trying to show the DNA as being tightly wound versus more loosely wound, okay? So we have tight DNA with torsional strain, and now we have more relaxed DNA. So topologically, topology is a branch of mathematics that deals with structural features, like the structural details of a coffee cup, for example. Topologically, a coffee cup and a donut are the same topological structure because they each have a single hole in them, okay? So that's a whole branch, a complicated branch of mathematics that we don't need to go into, but that's essentially what's happened is the topology of the DNA molecule has changed in going from this torsional strain to the more relaxed one. And I'm not sure why my pen color keeps changing here. I'm probably bumping something. Anyways, the topological features are changing, and that's why this is called a topoisomerase. All right, so now we have one more thing that we need to mention before we get into the actual replication part, the addition of new DNA nucleotides. Because when the helicase melts the DNA and turns it into a single strand of DNA, and then it moves along like you see between the first bubble and the second bubble. Notice what is keeping the DNA melted. Once the DNA helicase moves out of the way, what's to keep the two DNA strands from reannealing, like I'm showing here? Okay, because they're certainly complementary. We know that the DNA, the reason the DNA is double-stranded in the first place is because the DNA, the nucleotides are complementary and they base pair strongly with one another. So why wouldn't they just reanneal? Why wouldn't that happen? Or zip back together or turn back into double-stranded DNA? Well, we need something to prevent that from happening, because in fact it would. So there's another protein, which I'll represent with green, I think it is. So green proteins attach to the single-stranded region. So after the helicase moves along, these proteins will attach, and that will prevent the DNA from reannealing. So those are called, I'm not going to write out the whole thing. These are called single-stranded binding proteins, SSBP, single-stranded binding proteins. Okay? And there's multiple of these proteins that attach. They obviously have an affinity for single-stranded DNA, and they will stay in position until other enzymes come in and make the copies, make the new DNA copies. All right. So that's essentially the setup for DNA replication, the helicase, the gyrase, and the single-stranded binding proteins This creates the bubble, stabilizes the bubble, releases the torsional strain, so that we can now begin the process of making new DNA. So I'm going to stop this video at this point because we're about 20 minutes, and I'm going to do a part 2 video talking about the actual replication process.